Um, I think I'll start by letting you guys know a little bit more about me. I actually worked for Hoffmann La Roche between 1984 and 1990 in Switzerland, working on HIV. We left Switzerland and came to Cleveland, and I worked at university hospitals, again, working on HIV, and I established a Center for AIDS Research. And in 1999, we moved down to Frederick. I joined the National Cancer Institute. And the work in my lab has involved research on HIV, on Zika virus, on Ebola virus, on dengue virus. I mean, all the things you don't want to hear about. That's the sort of research we've been doing. And by sheer coincidence, I'm actually retired now. I retired at the beginning of the year. But as far back as about 2019, I was advocating for the National Cancer Institute to set up a program in mRNA vaccines. I also, once I retired, we'll get to this later about what is really the simplicity of an mRNA vaccine. One of the things I'd like to do in my retirement is teach, possibly at Poolsville High, and teach technologies like this. This is not, as I say, rocket science. This is very straightforward. It doesn't just apply to coronaviruses. It applies to all sorts of diseases. So I want to go over that with you for a part of my talk. And the other reason I wanted to give this talk is, as a scientist, it's been hard listening to the media saying that our science is not true. We can't be trusted. As scientists, we go in to do an experiment with a completely open mind. We make a hypothesis, we do the experiments. If it works, that's great, we get to publish the work. If it doesn't work, we're a little bit depressed, but we sit down and say, why did it go wrong? And see if we can correct it. And that's how we do our science. We do not go in prejudging anything. And so it's been hard for me for the last couple of years to see scientists really disrespected by many people in the media. And I do think we have to have our voice and try and dispel what really are a lot of untruths and myths and so on. So that was my work up to it. And the first slide actually shows you something that is really quite remarkable. And this is this Operation Warp Speed, where we actually turned what is normally 10 years plus of research into 10 months of research. This is absolutely unprecedented. And again, it's one of the criticisms is it went too fast. There are reasons why it went so fast, and we'll get to those later, but it is an absolutely, never in the history of vaccine production has something like this happened, that we've gone from the disease, the pandemic, to vaccines, and only a week ago, new therapeutics in less than a year. It really is remarkable. Okay, well, that's the first. We've just hit the first glitch. Just hang on. Let me see. Okay, so I am not advancing here. Kirsten? Just hang on. I'm not advancing. My technical assistant is here. Okay. That's the great thing about having your daughter here. All of the technical things get sorted out. Okay, so anyway, just to put some numbers to this, and I think we've seen things like this. There's the world situation, the global situation. There's 233 million people have been infected, almost 5 million deaths, 210,000 recovered. And on the right-hand side is the current status in the US. We have over 700,000 deaths. And I thought it was important to put that in perspective to see that during World War II, 405,000 American lives were lost. 58,000 in the Vietnam War and 36,000 in the Korean War. And so we've surpassed all of that with COVID-19, and that's a very sobering fact. So where are we with this virus? This graph, I don't know how well you can see this, but it's to depict pandemics through history. Okay, and you can see the Black Death is clearly the winner in pandemics. The numbers, over 200 million people died from the Black Death. We are down here at the moment, or we started down here with COVID-19. But what's kind of frightening is if you follow the red line, we've moved to about here with the number of deaths in the world at almost 5 million. And clearly that number is going to go up. This is the one we're probably most familiar with, HIV, with somewhere between 30 and 35 million people dying. But it really is quite frightening to see how this pandemic has taken off throughout the world. Again, another really sobering fact is that COVID-19 is the #1 cause of death in the US in 2021. And these are some figures for you to look at. Just a few minutes before my talk, I went online. I constantly check the COVID USA website just to see the number of new infections, number of deaths. And this is October 6th, yesterday. There were still 2,522 deaths a day. And at the peak, I think if you remember, we had about 4,000 to 4,500 deaths a day. We had a September 11th virtually every day. That's the bad side of it. The good side of it is that this is, again, it's looking at this compressing the timeline of 10 years for drug discovery into 10 months to let you see what's happening all over the world. Okay, so globally, there's about 190 experimental vaccines out there at the moment. These whittle down to what were called phase 1, phase 2, and phase 3. These are different phases of clinical trials. And underneath, it explains what each phase of a clinical trial is. I want to get back to this later because one of the reasons the development of the COVID vaccine was accelerated was because these phases were not done one after the other. Once phase 1 was started, halfway through that, there was enough confidence in the data that they could start phase 2. Once phase 2 was moving, there was enough confidence they could start phase 3. So in essence, they overlapped everything. And by doing that, they saved a huge amount of time. And so the end result of that was that we actually have 21 vaccines at the moment. We have the Moderna and the Pfizer vaccine, the AstraZeneca, the Johnson & Johnson. Russia has several vaccine candidates. China has several vaccine candidates. India has vaccine candidates. And of these, about 8 are being monitored in the wider population after being approved by the regulated authorities. So again, we've whittled this from 194 potential vaccine candidates to 8 vaccines that are actually being used in the wider population. They do have approval. And again, I can only, it's almost jaw-dropping the speed that we can do something like this. So the next thing is it's the start of some of the myths of COVID-19, okay? So the vaccine is an RNA vaccine. And this is a little bit of basic science. This is what we call the central dogma at the bottom of the slide. DNA is in the nucleus. DNA in the nucleus gives rise to RNA. The RNA then leaves the nucleus, and in the cytoplasm, it makes protein. And in general, you'll see the direction of flow is from left to right. DNA makes RNA, RNA makes protein. And this is important because there have been several misconceptions that the RNA vaccines can get into your DNA. And I'd like to state unequivocally that this does not happen. The direction is DNA to RNA, and there is no way for the RNA of this vaccine to go back into the DNA. The direction is forward for the cells to make protein. So what does the RNA vaccine do? There's a picture, a representation of COVID-19 there, a very simple representation, and it's actually a very simple virus. It doesn't have many components, but the most important one is what's called the spike protein. The spike protein is on the outside of the virus, and if you follow pathway A, the spike protein then attaches, once the virus attaches to the cell, the spike protein allows the virus to fuse with the cell, and the RNA of the virus enters the cell. On the other side, what we do is with an mRNA vaccine, once the messenger RNA is inoculated, our cells themselves make the protein, and the body then has a response to that protein by making antibodies. And that's the basis of the vaccine. The spike protein is the first protein that your immune system would see. And so that's obviously the best candidate for making a vaccine. Now, down at the bottom right is really the big problem. The virus, when the virus is allowed to sort of replicate, you might say unchallenged, the virus is not really precise at duplicating itself. It makes mistakes. It makes them all the time. But a lot of the times, the viruses that have those mistakes die. They can't survive. But now and then, one gets through, and the mistake in the virus, it can live with it. And the Delta variant is one of those. The Delta variant has changed a tiny, tiny part of the spike protein, and that is enough to make it more infectious. And this is actually extremely important later on when we're talking about trying to engender herd immunity and things like this. The other thing is the Delta variant is what's circulating in populations that aren't vaccinated at the moment. And for me, a real concern, if we don't really put more effort into our vaccination program, is as the virus is allowed to replicate and survive, we're going to uncover worse than the Delta variant. So again, I think it's extremely important that we push the vaccination program as hard as possible to make sure very few variants get through that actually will resist the current vaccines that we have. So what are the advantages of a messenger RNA vaccine? The first is their safety profile. And again, this goes back to the notion that someone had said that the RNA gets back into your DNA. What's called messenger RNA is very short-lived. And because of that, it will not be inserted into your DNA. The other advantage of the messenger RNA vaccines is that we don't use any viruses to deliver them. The vaccines are incredibly simple with the RNA and a little bit of fat around it that helps them get into the cell, and a little bit of salt and a little bit of sugar, but they are incredibly simple. And because the vaccines, these mRNA vaccines are simple, manufacturing them is quite easy. They can be scaled up. In my lab, As I said, the last research we did on my lab making mRNA vaccines, these were against cancer. We worked at a scale of RNA that was, for our purposes, enormous. The scale that we're talking about with production of mRNA vaccines is probably 1 million to 10 million times larger than that. The other thing is because the production is carried out completely outside a living organism. This is all done in test tubes in what we call bioreactors. It's like taking the test tube that I have and increasing the size of that 1 million-fold, okay? Because of that, we really can turn out hundreds of millions of vaccine doses in a matter of weeks. So that is one of the real advantages. The other advantage is what I've written in red here, is the technology is so simple that if variants of the virus come up and we need a second generation vaccine, this can actually be done in a matter of weeks. The technologies we have nowadays would allow us to decipher all of the genetic information of a new virus. Let's just call it a new virus. Within days, we can get that information. Within days after that, we can go back to that part of the virus we were using as a vaccine, but now it's altered in the new virus. Within days of that, we can have the first sort of early phase vaccine, not for clinical trials, but for trials in the lab. And so this is something that is really important. And this is something we can't really do through normal vaccine research. The response to a change, if your vaccine is not working because of the development of a resistant virus, getting to a second generation vaccine by standard methods is a little bit slower. Whereas here, it's weeks. Within weeks, we would have a second generation vaccine, and I'm quite sure we have second generation vaccines. It would not surprise me if we have vaccine mixtures relative to the original vaccine, to the Delta vaccine. But that's what's possible with mRNA vaccine technology. And the other thing, as I referred to this a little bit earlier on, the mRNA can be customized to the target. We're talking today about COVID-19. As I said before, the last thing we did in my lab is make an mRNA vaccine, a cancer vaccine. The rules, the disease that you're targeting, regardless of the disease, the rules for how we go about it don't change. We target a little bit of RNA, we make it in huge amounts, we inject it into the cell, and we look to see if the cells produce an immune response. And this is something that some of you may have heard before, but using a technology like this and the ability to manufacture the vaccine in huge amounts and quickly opens the door to what we call personalized medicines. This has been a very popular theme over the last 10 years. And now we see with mRNA vaccines and the success of the COVID vaccine that we can turn this now to other treatments. Now, this is getting to another misconception that's out there about the vaccines. People are saying it's very new. As a scientist, that doesn't bother me. What is more important, is it safe? Is it reliable? But what I put up there were just 4, these are manuscripts taken from the literature showing that, and these go back to 2017, just showing you that mRNA vaccines have been considered for Zika virus, for rabies, for HIV. And in actual fact, the therapeutic RNAs date back to about 1990. So the field is actually 30 years old. Now you might ask, why, if it's 30 years old, why haven't we had therapeutic RNAs or messenger RNA vaccines 20 years ago? And this is actually there's a very simple reason for this. It's not that we can't make RNAs to make vaccines. It's that at the time, the technology to scale up the production of the RNA wasn't there. And it's taken us this in-between phase to understand how to make an RNA vaccine, how to prepare the RNA that it doesn't break down when it goes into the cell, how to prepare the RNA. As I said, we worked in microgram amounts of RNA in my lab. Companies like Mrna and Pfizer now make this in 10 to 20 kilogram amounts. RNA production at this level was unheard of in 1990. And so it really is a simple matter of the technology to scale up. And if we think about this now with the COVID pandemic, with 8 billion people in the world needing 2 shots possibly, and let's just say there's some wastage, we're talking about needing the RNA to make 20 billion inoculations. And we have not, until the last 10 years, had the practical technology to do that. Now we do, and I think we're going to see in the coming years, there's going to be more and more messenger RNA vaccines. Is the vaccine safe? Okay, this is, the vaccine is, in my opinion, is as safe, or the COVID vaccine is as safe as almost any other vaccine. I am not going to say there are no problems with it. There are side effects in certain individuals, and we have to be very careful with those individuals. But I wanted just to put this slide up. When we talk about safety, of the COVID vaccine, and these are some figures that I pulled from the web about some of the over-the-counter medications that we use. NSAIDs like ibuprofen, 16,000 people die a year. Opioids, 14,000. Aspirin, 8,000. Acetaminophen, 550. So nothing that you might say, nothing foreign that we put into our body comes without any potential side effects. I'm not trying to downplay the side effects of a COVID vaccine, but I think we have to realize that some of the side effects, the rates that these are turning up are, they're single-digit numbers per million. And I don't think there's been one complication of the COVID vaccine that has really been a significantly large number of people. So there have been multiple clinical trials. There are the occasional myocarditis in young children and young boys is a problem. And as I said, we have to be careful with these and look at other ways of treating these individuals. But in my opinion, the COVID-19 vaccine, we were at CVS last week. I got COVID in the left arm and flu in the right arm. I regard them both as safe. The one as safe as the other. Okay, we want to go on to some of the myths of COVID. It's been said that the vaccine can affect women's fertility. Put quite bluntly, we have only had a vaccine for 10 months, and there is simply not enough data to suggest or to support the notion that it affects women's fertility. There is no reason why the vaccine should affect a woman's fertility. This is like any other vaccine. The vaccine encourages the body to make copies of the protein found on the virus surface. It makes antibodies. The antibodies then attack another infecting virus. So I think it's safe to say that the COVID-19 vaccine will not affect women's fertility. So another question is, if I've had COVID-19, do I need a vaccine? The answer there is yes, because what you might call natural COVID-19 infection, your body raises antibodies, but these tend to wane and decrease in their potency rather quickly. And so if you're recovering from COVID-19 infection, you may for a couple of months have a reasonable amount of antibodies, but these are not long lasting. And for that reason, I think it's very important to get the vaccine, and for those of us who are eligible, the booster. It's the best method of protection, whether or not you've had COVID. Vaccine development was rushed, so effectiveness and safety can't be trusted. This is the point I brought up in the very first slide. The vaccine, normally the clinical trials would, as I said, would be done step by step. You would do a phase 1 clinical trial and you would sit and look at the data, decide whether it is worth going forward to phase 2. You would then set up a phase 2 clinical trial, wait till the end of that and see how the results were, and so on and so on. Here, the vaccine producers and those involved in the clinical trials decided halfway through one of the phases that the results looked good enough to start phase 2. So you had a phase 1, phase 2, and then downstream of that, phase 2, phase 3. And that really cuts months to years off the time it takes to get approval for a vaccine. Also, because I'm talking about the mRNA vaccines, there was enough confidence after the phase 2 and going into the phase 3 clinical trials for the pharmaceutical companies to start producing the vaccine. Normally, the conventional method of preparing a vaccine, that would come far later after several regulatory steps. But the pharmaceutical companies this time decided this looks good enough and we can start manufacturing the vaccine. It was a risk, but in my opinion, it was a risk that paid off. The other thing is that Operation Warp Speed pushed a huge amount of funds, government funds, into this. And so we didn't have to give out, let's say, grant money to institutions. They didn't have to spend a long time writing applications for the money. The government moved very, very swiftly. The other thing is the government paid for the vaccines in advance. And again, since vaccines are prepared faster than traditional methods, clearly the effect of that, the development was faster. So nothing has been missed out in terms of safety of the vaccines. And we have administered now, I think, globally, 6.5 billion vaccines. The data still looks very good. And so, again, in my opinion, their effectiveness and safety, I think, has been proven globally with these 6.5 billion vaccinations. Okay, this is a word, we've had several discussions of this with our friends, and there's sometimes even we have trouble explaining this issue of vaccine efficacy. How efficiently does the vaccine work? The main companies I've put down here, the numbers there, for example, Pfizer, their vaccine is 91.3% efficacy. It's 88% effective against the Delta variant. The Mrna is 90% effective against infection. Going down, the Johnson & Johnson has 72% overall efficacy and 86% against moderate disease, severe disease in the US. You'll see these numbers are going up and down, and they're ranging between 100% for the AstraZeneca And in some cases, it goes down to about 60% for AstraZeneca. I think we have to understand that the 2019-2020 flu vaccine was 45% effective. And normally the flu vaccine is somewhere between 50 and 60% effective against influenza A and B. So the take-home lesson of this is if you look at all of these numbers, they are all good. Every vaccine, whether it's an mRNA vaccine, whether it's a viral vaccine, they are all good, despite these efficacy numbers ranging from 100 to 60%. So another issue that was thrown about, that was brandished, was could we reach herd immunity? Okay. So on the left-hand side, you see in the absence of herd immunity, there are susceptible and infected people around, so you basically pass the virus on to your partner. Once you have the herd, you see that susceptible people are separated from each other by immune people. Now, herd immunity is more theoretical than practical. In fact, it was tried in Sweden and they stopped the trial because they could not reach herd immunity. And if you remember going back about a couple of years, one of the president's advisors brought up the notion that maybe we should try and go for herd immunity. Let's go for natural infection and get to herd immunity. That notion would have cost 2 million lives in the United States. So that to me is something that is totally irresponsible to suggest that we just let this virus run wild and eventually we'll reach herd immunity. The other thing is that sad fact is we aren't going to reach herd immunity. The reason for that is it's this thing in red in the second paragraph. I have to do a little bit of science here. This is called the reproduction number, or R0. For the original virus, R0 was between 2 and 2.5. Now with the Delta virus, that means that every person who was infected would transmit to between 2 and 3 people. The Delta variant, the R0 number, is somewhere between 5 and 8. It is much more infectious, and so you pass it on to many more people. So in order to reach herd immunity with the Delta virus, we need to get between 85 and 90% of the population vaccinated. When we talked about herd immunity earlier with the original virus, we were talking about somewhere between, say, 60 to 70%. That changed immediately with the arrival of the Delta variant. As I said, we need between 85 and 90% of the population needs to be vaccinated. And you see the last figure, only under 60% of the US population is vaccinated. So the result is that achieving herd immunity is going to be either unlikely or long-term. But what's built into this, which makes it even a little bit more frightening, is for as long as we have a population in the country that are unvaccinated, the virus is still allowed to grow. We've heard of the Delta, we know of the Delta variant, there is the Mu variant. There's undoubtedly going to be other variants that possibly will evade some of the earlier vaccines. And this is this frightens me, that until we get this under control, if the virus is allowed, as a virologist, if you let a virus reproduce and replicate uncontrolled, the virus will turn up resistant variants. It'll turn up more dangerous variants. We've noticed now, we've always said that children don't seem to be as susceptible to the virus. And one reason is that the cells that the virus attacks in children, they have fewer of the receptors that the virus can attach to. Well, yes, that's true. However, the Delta variant, that doesn't bother it because it can attack where there's even fewer receptors. And this is, again, what is really quite alarming, that we're seeing children, the infection rate in children is starting to rise, and it's rising really alarmingly. And we're hearing terms like long-term infections in young children. And I think as a parent, this is something I would be extremely concerned about, and I would be really pushing to have my children vaccinated, because the virus, it's not a virus for adults. It's not a virus for children. It's a virus that will attack whoever is not vaccinated. Okay, the alternatives to the vaccine, you may have heard about this, are so-called monoclonal antibodies. The company Regeneron has. The monoclonal antibody is basically what it says. It's an antibody. Your body doesn't have to make it. It's already been made, and it's injected into the body. Okay, it is very effective. However, the company Regeneron is selling the cocktail to the government at $2,000 a dose. Now, it is free to those who need it. Two things here. It costs 50 times more than a Pfizer vaccine. And this $2,100, these are your tax dollars, okay? So this is expensive. The public is paying for this one way or the other. The other thing about the monoclonal antibodies, they work almost immediately. However, the protection only lasts for a few weeks to a few months, meaning you can be reinfected, meaning you'd have to go back in, get another antibody, monoclonal antibody therapy. There's a practical issue of this as well, that the monoclonal antibodies must be infused, okay? And there's a little antibody infusion tent below. So this is not simply, it's not the equivalent of going to CVS and going behind that little curtain and getting your antibody. You have to go to an infusion tent, you have to have the antibodies infused. This all has to be done under sterile conditions. And you can imagine, this is also a real logistical challenge, using the monoclonal antibody therapy. There are certain people in the population that can't be vaccinated, and for those people, clearly, monoclonal antibodies are the solution. But this is not and should not be for the general public. It is simply so much easier to go down the road to your CVS, roll up your sleeve, and get vaccinated. You may have heard recently, I'm not going to talk about the other drugs. Remdesivir, we've heard about, but what I call the other drugs, that the dark side of the COVID drugs. I don't want to talk about those. More recently, Merck has developed an antiviral drug. And basically the drug, it tricks the virus into making mistakes in its RNA as it multiplies, and that kills the virus. Now that's the glass half full. And having worked for 20 years with HIV, what's going to happen is the minute you challenge the virus with the drug, the virus will start to develop resistance to it. That viruses they have for such a small amount of genetic information, the viruses are very clever. And I think that we're going to see the emergence of drug resistance, regardless of the therapy, the monotherapy that's being introduced by Merck. In HIV, we've made enormous progress. Is we don't use a single drug. That's what we call a monotherapy. We use a cocktail. We now use a cocktail of 3 or 4 drugs. And that makes it more difficult for the virus to escape. Hang on, my computer is asking me to do several things at once. But I think it's important to know that we are making progress along the lines of COVID therapeutics, but I think we have to be a little bit cautious in terms of our optimism there. Okay, so I said I'm going to end on the more controversial issue of COVID-19, and this is the use of human cell lines. The AstraZeneca vaccine and the Johnson & Johnson vaccine, these are prepared in what are called HEK 293 cells. The HEK stands for human embryonic kidney cells. These are cells derived from fetal tissue in 1970. First of all, I want to say there is no fetal tissue in any vaccine that's been administered at the moment. HEK 293 cells, these have been used for the best part of 50 years to manufacture vaccines. These are preferred over other non-human cell lines because they tend to grow better. The virus grows better in these cell types. They can be maintained, and you can use cell lines from decades ago. But we have to be clear here, it is a cell line. The cell line of 2021 probably doesn't bear much resemblance to the cells that were taken from the fetus in 1970. The vaccines don't contain any aborted fetal cells. The Pfizer and Mrna vaccines haven't made use of fetal cell lines. Now, that being said, in defense of using human cell lines, I think it's the community, the scientific community, I think their eyes have been opened to the sensitivities of people. This is not something we should just wash under the carpet and say, this is how we do it, and this is how we'll keep doing it. It is possible to look for alternative cell lines that are not as controversial, and I think the onus is on the pharmaceutical companies to do that. But just to let you know how modern medicine has benefited from the cell lines, these are two lists of over-the-counter drugs and prescription drugs that in one way or the other, they've been tested on the same cell line. And there are several things here that many of you are conversant with: Tylenol, Advil, loratadine, blood pressure, atorvastatin for your cholesterol. So this cell line, modern medicine has really benefited from the use of this HEK 293 cell line. Also, therapeutics that have been produced in HEK 293 cells. I won't try to pronounce the things on the left, we'll go more to the right, but therapeutics for septicemia, hemophilia, type 2 diabetes, rabies infection, rheumatoid arthritis. These have been produced in HEK 293 cells. This is my last slide, and for me, this is something very important. When the pandemic started, I talked with many friends about the logistics of a vaccine campaign. Let's talk about America, and then let's talk about the world. We're looking now at two vaccinations, two shots, Pfizer or Moderna. We have a US population of about 360 billion. That means we need 720 million vaccine shots. Now let's say there's 30% wastage. That's 1 billion vaccine shots we need in this country alone. Now let's do that globally with a population of about 7.5 billion. We need somewhere of the order of 20 billion doses of vaccine. That's number one. Can we actually rise to that? Can we provide just the raw materials to make 20 billion vaccines. I think we can. I think the technologies that I've been talking about tonight, they're being constantly improved. But there's a really important thing here, and this is the supply chain. I don't know if you saw I think it was on CNN the other night. In the US, we're now running short of cotton. There's several things we're running short of. We know about the semiconductor issue with cars. And the important thing that we have to understand is that unless the entire world is vaccinated, countries way over on the right-hand side of the globe there, who produce things that America on the left-hand side needs, until their COVID problem is solved, the supply line breaks down. And every link in this chain has to be taken care of. And so I think it's extremely important that while the US, while the EU is moving forward very rapidly in getting the population vaccinated, there is a real responsibility on us to make sure that other countries in the world, in the East, in Africa, make sure they are also vaccinated, because we do, one way or the other, rely on them. And until you see here that some of the countries won't have 60% of their population vaccinated until late 2022, 2023. As you can imagine, if these countries are producing things that are needed in materials sold in America, we are going to be spinning our wheels for a year or so. So global vaccination is something that we really have to give serious thought to, not just America, the developed countries, the EU. We all need to chip in here and make sure that the less fortunate parts of the world actually get access to the vaccine. So that was my last slide. And I'm not quite sure how I get, if I stop sharing.
We can go ahead and open it up to questions now.
Yeah.
I see the first question in the chat says, do you think that future flu vaccines will make use of the mRNA technology?
I can't say. I think the current flu, the thing is, if we made it through an mRNA technology, the flu virus mutates so quickly that the mRNA vaccine we make today may not work against the next flu strain. This is the problem with flu, that every year there's a new strain. What one could do is look at the flu strains, let's say over the last 10 years, and pick out the protein that we're going to make the mRNA vaccine. You could actually inoculate with a cocktail of vaccines, mRNAs, against different flu strains. But I think that runs the same risk, that flu mutates so rapidly that probably on an annual basis, we would be making new vaccines.
We can open it up now to any other questions. Feel free to either send them in the chat or unmute this time and ask them yourself.
Yeah, I signed up for a booster, and the paperwork that they sent said I would either get a Pfizer or a Comirnaty. What is that? Is that like a generic?
I think that's the generic name of it.
Okay. All right.
There's nothing mystical about that. I think that is just the generic name of it.
Okay, I just never had heard of it before.
Well, it's easier to call it the Pfizer vaccine.
Okay. All right. Thanks.
Stuart, right now the spike protein is what causes the mutation, right?
Yeah.
Is it possible that at some point the actual virus itself, or the nucleus of the virus, will also mutate? Is that something that happens in viruses?
Yes. We get a little bit more technical here. The
Sorry.
proteins on the inside of the virus, the nucleocapsid or the capsid, these all have to fit together in a very, very organized way.
Okay,
So it is, if there's a small change in one, if one amino acid changes, this could sort of set off a chain reaction throughout the whole nucleocapsid. I don't think I think it's not unreasonable to expect that mutations turn up. I said at the beginning, there are some that the nucleocapsid will just fall apart and the virus will die.
Right.
There's others where it may not replicate as well, but it'll survive that. I'm not sure if that's quite the answer you want.
no, that helps me, yes. And also to follow up, is the SARS, this is SARS-CoV-2, is it related at all to SARS, the original SARS virus?
They are all related. They're all coronaviruses, SARS, MERS.
Right,
They're all related. Yeah.
Okay, all right.
I'm curious what you think about school policies. I know I'm at the University of Maryland, and this semester we're back on campus, and they're requiring vaccinations for most students and faculty, of course, without any of the medical exceptions. But everyone's still kind of been very iffy about whether or not that kind of thing is actually a safe decision or will last.
Getting vaccinated, having a vaccination policy.
Well, having all of the students on campus in such a dense area when especially a lot of the surrounding county has a pretty high COVID rate.
I think my answer to that might not be the answer you want, but my answer to that is I would like to see the vaccination rate in the students rise. I think we're going with this vaccine or with this pandemic, we're going to have 3 layers. Think of it as a chocolate cake. So the upper layer of the cake are those of us who are vaccinated, the 60%. The lower layer of the cake is those of us who are going to get vaccinated, and that's maybe 20 to 25%. And the icing in the middle is the people who will never get vaccinated. And I think that's about the best we can do. That, you know, as students are, once the students are vaccinated, I can't guarantee, but I would not be too concerned in that environment. I think the students are probably more responsible than other parts of the population. I think they understand the need to be vaccinated.
So it sounds like from what you're saying about this herd immunity that we are never going to achieve, that we just have to get used to COVID being part of our lives.
Yes, it will.
And
it'll be endemic. It won't be a pandemic. It'll be endemic.
right.
The virus will come to grips with us. You know,
Right.
it's not the cleverest thing for a virus to do. To kill you is not the smartest thing for a virus. You want to live with your host. And so, you know, we and the virus are just getting used to each other. So eliminating it completely, I think the probability of that is pretty close to 0.
Right.
I think it being endemic and getting our COVID shot as well as our flu shot, excuse me, that's what we'll be looking at in the future.
Okay, it's probably like a yearly COVID vaccine similar to flu, you think?
I would imagine, you know, where there's much of this, you know, it's a lot of the questions that are being thrown out there. It's sometimes difficult to get answers because we've been 10 months into this.
Yeah.
And we don't have that sort of data yet. How long will the booster last? I mean, boosters only started a month ago. So we're learning as we go along. You know, the good thing is that the effort we put in from pharmaceutical companies, you can really see that there is a huge amount of effort and basically goodwill in this. But yes, I think we'll be looking at boosters on an annual basis.
Okay. All right. Thanks.
Okay.
And do you think that will be eventually boosters for everyone or just for certain segments of the population?
I would like to think it'll just be for the more susceptible of the population. The problem is, as I said earlier, we always thought this was it was older people who were the problem, and then we realized that it was people from sort of 40 to 60, and you know the age group is going down and down and down in terms of who can be infected and the severity of infection. I would imagine children under, let's say under 12, I might set that aside from the rest of the population, but for the rest of the population, I would think that booster shots every year are going to become the standard.
I see another question in the chat that says, one of the slides you showed deaths from aspirin, etc. How many from the COVID vaccine?
Deaths from the COVID vaccine. I would have to look that up. But I think I can say fairly safely that it's less than 12,000 this year.
Yeah, from a quick Google search, it says around 5,000 something.
Yeah. I think one thing when we talk about safety of the COVID vaccine, many of us have seen adverts on the TV for the latest drugs for disease X, Y, and Z. And it turns out when that advert, it takes about a minute to explain all of the side effects of the drug than the benefits. And this just makes me think that the side effects of COVID are, in my opinion, with a few exceptions, are not severe. They're as severe as the side effects from many of the over-the-counter drugs that we take, prescription drugs that we take. The fatality rates are comparable, and so it's no better and it's no worse, I would say.
Thank you. Do we have any more questions either in the chat or now is a good time to unmute? I'm curious if, in your opinion, there's anything systematic wise that's holding back from getting a lot of our population vaccinated. I know a lot of people are making that choice just based on personal preference and opinion, but on the side of the system actually distributing that vaccine, where are places that you think maybe could be improved?
That is a really difficult question to answer. You're talking to a scientist who, when I traveled to China, went for all of my vaccinations, and when I traveled to Africa, went for all of my vaccinations. I've never seen vaccination as a hurdle. And so I don't know where these people are coming from. I think a lot of it is misinformation. I think the hesitancy is we should not read Google and the internet until we get the answer that we want. We should read and educate ourselves and look at the pros and cons. And I'm concerned that a small percentage of the population is reading to get the answer that they want, and they're being misinformed by people who really have no authority and background on COVID, on the epidemiology. And I think more open discussions like this, I think community leaders, I think in churches, this is really important, doctors talking with their patients, because we have had for a while a misinformation campaign. And this has to stop because the virus is going in the direction of young children. Excuse me, and as a mother, that would worry me terribly. to be told that, you know, the virus is unsafe, that it doesn't do this, it does this and that to you. Well, there's a possibility of your child becoming a so-called COVID long hauler because you've listened to other people. It is now down to very personal decisions, and I have made mine, my wife has made hers, our daughter has made hers, and I really don't know what to say to some of the people. For example, people have said, I need to wait a bit longer. And my response to that is, define longer. When 2,500 people are dying a day, how much longer do you have to wait to find out if this virus is safe? And what do you mean by longer? What do you mean by safe? And these to me are just, they're statements that people just throw out. I need more information. What more information do you need? We've inoculated 7 billion people. The statistics are there, the statistical power is there. And so it's difficult sometimes to have a candid discussion when someone throws that statement out at you. I need a bit more time. 2,500 people this year could have done with a bit more time. They died of COVID.
Thank you. That was a very thoughtful answer. And I also wanted to address in the chat, I see Tina, you asked if this will be available later. And yes, this was live streamed on our Facebook, so you'll be able to see it there. And also it will be up by this weekend on our YouTube. So if you're looking to share this resource, you will have a link, and it will also be posted on our blog and Facebook. So it'll be all over our pages. You can get it there and send it along to anyone you would like. Do we have any last questions? Well, I just wanted to say, I hope you all enjoyed tonight's presentation and learned as much as I did. If you think of any questions later, you can always email us at info@poolsvilleseniors.org and we can pass them along for you. If you'd like to unmute or turn on your camera to say goodbye, now's the time. And if you're joining us on Facebook Live, please leave a like and a comment. We'd like to thank Stuart for this presentation. We couldn't have had a more knowledgeable presenter this evening. You shared so much knowledge with us, and it was so in-depth, and I really, really appreciate that. We'd also like to thank our ongoing sponsors and private contributors, which help us keep our programs going because we love putting them on for you. If you enjoyed tonight's program, make sure to check out our website and register to join us for more upcoming events. Like I mentioned at the beginning, this time next week, we'll be back with Paranormal Poolsville. And you can find all of that at poolsvilleseniors.org for more info. And like I said, this link to this presentation will be up there this weekend as well. Thank you all so much for joining us tonight.
Thank you, Stuart.
Thank you,
Thanks,
Stuart.
You're welcome.